Our gene is JAG1, which creates jagged1. It also creates a cell signaling called Notch signaling. It is included in 13 different tissues, including brain, lungs, pancreas, and other tissues around inside the skin. Notch signaling also controls how certain kinds of cells develop in embryo, including the liver, heart, eyes, ears, and spinal column. The locus of the gene is 20 chromosomes at position 12.2. Nucleotides is 6,048 base pairs long, and expression in other cells is, like I said, 13 different tissues.
The three processes of transcription are initiation, elongation, and termination. During initiation, RNA polymerase binds at the promoter and unzips the gene. During elongation, it reads the template from 3' to 5' and builds an RNA complementary strand from 5' to 3'. During termination, the last process, it reaches a terminator and stops. So you can see how the RNA polymerase comes down and elongates this, and then how it reaches the terminator and stops. And then you can also see the RNA strand being built. The location of it occurs in the nucleus, and the purpose of it is to make mRNA from DNA. RNA differs from DNA because RNA is single-stranded, not double, and it also has bases C, G, A, and U, not C, G, A, and T. Also, in RNA, ribose sugar is the sugar, and in DNA, deoxyribose is the sugar. mRNA processing occurs to make sure that the ribosome recognizes the gene and so that the sequences do code for the protein. The 5' cap allows the ribosome to recognize the mRNA sequence and to bind to the mRNA when translation begins. The poly A tail located at the 3' makes it known to other organelles to not eat it, and that it is an important part. The difference between introns and exons is that introns do not code for the protein and exons do. So what happens is the introns are removed during mRNA processing and the exons come in, so it makes mRNA.
For translation, the purpose of translation is to assemble amino acids into a protein chain. It is located in the cytoplasm.
The ribosome, made up of RNA, moves along with mRNA and reads the codons. tRNA comes in with anticodons to match up with the mRNA codons. Thus doing this, amino acids are formed. Then, later in the process, amino acids form peptide bonds through polymerization. Finally, to end the process from going on to infinity, a stop codon is needed to end the protein chain.
Codon tables can assist scientists in finding the amino acid sequence because you can put the codon that you already know into the table and figure out what amino acid it will become. In JAG1 for mutations, 226 mutations have been found or discovered inside of JAG1. There is two main types of mutations. A Legel syndrome, I think it's how you pronounce it, is the loss of JAG1 protein, which is a lack in Notch signaling. An error in Notch signaling can lead to an error in development of parts of the human body. The other main mutation inside of JAG1 is cancer, which is the increased activity of JAG1. An increase in activity can lead to tumors such as head and neck tumors and breast cancer. Increased expression also can be a cause of cancer. This may lead to the promotion of blood vessels that can nourish or grow the tumor and enhance other cancer-related events, including cell division or proliferation and the inflammatory response. Thus, this is the end of our JAG1 sequence. That is all the information we could find. JAG1 can prove very useful of a gene, but it can also have many mutations added into it. Thank you for watching and listening.